guys. I hope everybody is doing okay. We're going to do some cell reproduction notes, so just mitosis. We'll look at meiosis later, but we're doing this so we can get into our genetics unit. So cell reproduction, you can kind of see it up here. Hopefully you remember a little bit, maybe from biology 1. So let's go ahead and get started. Just a big science nerd joke. So anyways, starting off with that. I know everybody usually looks at me like I'm crazy with that one. So anyways, let's look at this. I'll just kind of highlight what you guys need to be writing down. So the cell cycle is just in orderly steps between eukaryotic cell divisions. So that's the big thing here, is that it's a eukaryote. This does not just happen, this doesn't happen in prokaryotes. It's eukaryotes. And what's the big difference between a prokaryote and a eukaryote? It is the nucleus, okay? Prokaryotes do not have a nucleus. So the cell cycle and mitosis in general is all dealing with the nucleus. That's why it's eukaryotic. So I will ask students, why do cells divide? And before this pops up here, and I get all kinds of answers, but the real reason why our cells divide is we grow, right? Even if I think I'm done, I'm done growing, like I'm as tall as I'm gonna get, like we grow in other ways, all right? So I'm constantly losing skin cells. Maybe I'm getting bigger, whatever, but you are growing. Reproduction, so in single-celled organisms, and it says that in single-celled organisms, they're constantly reproducing and splitting. They will literally go through mitosis to split and become two. And then to repair cells, we need to repair cells, which is kind of a good kind of jumping point from what we just did. We have cells that maybe were dying or damaged during immune system stuff, and so then we need to repair them or maybe get rid of them altogether and build new ones. All right, let me erase this line here. All right, control of the cell cycle. So this is something we do not discuss in biology 1. So during interphase, which interphase is where our cell spends most of its time. Now, I did tell you guys that in bio 1, but we did not talk about the parts of interphase, and this is it right here. So G1 is skip that. So we're going through, interphase G1, S, and G2. Sorry, I was getting ready to go into S, but it's not up here. All right, G1 checkpoint. This is the checkpoint to see if DNA is damaged, right? We need this. Could you imagine if we didn't have this? If your DNA happens to be damaged and you make a new cell with that damaged DNA? So this is really good that this is here. So your cell is growing, but it's also checking the DNA to make sure that everything is okay. The G2 checkpoint, and if you're looking here, G1, G2, it checks to see if DNA is replicated properly, which is also a good thing. So first off, that your DNA is okay, nothing has happened to it since you made that cell, and now that you're going to make another cell and you've copied DNA, which if you look here, this is where you're copying the DNA, so when you're done, you're now going to check that DNA and make sure that it is okay. Now, just because this happens, like this, I mean, think about it. For us, overall, we are pretty healthy people, but every now and then this can go a little awry, and stuff can happen, which we'll talk about. The M checkpoint, that is called spindle assembly checkpoint. It checks for the alignment of the chromosomes, so it's making sure that your chromosomes are okay. Okay, so a big word here. Apoptosis is a programmed cell death. So if any of these checkpoints fail, it will automatically, your cell basically kills itself. All right, and this is called apoptosis. It's a programmed cell death. All right, so let's move on. Let's go to much nicer topics, hopefully. All right, so mitosis and cytokinesis. So what we just saw there was interphase. Now, interphase, along with mitosis and cytokinesis, is all considered the cell cycle. So a lot of times people hear cell cycle, they think of just mitosis. It is all of this stuff together. All right, so what is divided during mitosis? It is the division of the nucleus. And that's going to result in identical copies of chromosomes. So the result in identical, complete copies of the chromosomes. Now that's what you're hoping for, during anaphase, what we'll see the phases here in a second, if they do not split, say that this chromosome here didn't split and go this way, say that it accidentally held on to this side and went this way, well then you can have a cell that has too many chromosomes. This one would have too many, and then you would have this one that is has lost a chromosome. This side has not enough. And so you have two cells that were made that aren't really great cells, but we see this. We see this in sex cells. That's how we have Down syndrome. And there's Klinefelter syndrome. There's all kinds of syndromes. Klinefelter syndrome, there's cat-eye syndrome. This is all due to these movements of chromosomes not happening correctly during anaphase. All right, so anyways, let's move on. We have cytokinesis, which is just division of the cytoplasm. That is the very last stage. That's the very last thing that happens during the cell cycle. All right, so here is this. We have interphase. So interphase looks like this, just a big jumbled up mess of your DNA. Prophase, you can start to see those chromosomes now, but look, you can see that nuclear membrane still. This will start to break down during this phase and into metaphase. It's gone now. But meta, middle, that's what I think of. And then you go from metaphase, you're going to jump over here to 4 to anaphase. I think of apart or away. And they're breaking apart. Number 5 is telophase, and I think of 2, T-W-O, right? You're breaking into 2. So there's a little bit. Prophase, I don't really have anything good to go with it. If you can think of something, let me know. And then 6, we have cytokinesis, which our 2 cells are now 2. The cytoplasm has split into 2. All right, so those are the phases. Make sure you've labeled them. All right, so down here, I'm just kind of wanting you to write down what is going on during these phases. So here, sorry, I kind of went quickly. Let me scroll back just a little bit. This is just kind of a chart that you can see. You can kind of see what's going on here. So they're showing you during interphase, and you can write down below your little drawing there this, but it's on the next slide too. So in interphase, you can see where there's a nucleus, there's centrioles, which we're going to talk about here in a second, a nucleolus, nuclear envelope. During prophase, we've got these spindles that are now forming. Look back in interphase, they're not there. Those spindles are now there. The centrioles are getting a little bigger. They're moving to opposite ends of the cell. Your chromatids, you can see how your chromatids have changed during prophase. Prometa, this is like this in between. You're not quite out of prophase, you're not quite to metaphase yet, but you can see the nuclear membrane is really starting to break down. When you see the word kinetochore microtubules, that is the spindle fibers. And then your nuclear, like I said, nuclear envelopes breaking down metaphase. These chromosomes are lining up down the middle, or what is called an equatorial plate. We're getting some scientific terms in here. During anaphase, if you notice, they're no longer calling them chromatids. They're now calling, they're calling them chromatids. Where is it? They'll call them sister chromatids, and then you'll get like a different word here. And then telophase lastly, and telophase and cytokinesis, where you get the splitting of the cell. All right, so let's look at this. This is going to show you a video of what exactly is happening during mitosis. You have 2 sets of cells there, so maybe this is an organism that only has 2 chromosomes. I mean, they're out there. We have 23 pairs, but this organism seems to only have 2 pairs, but it's still showing you exactly what is happening. There's a splitting cytokinesis, so here we go. They're forming chromosomes. You're in prophase, prometa, and here's metaphase, where they're lining up. You see the spindles come up. Anaphase. The nuclear membrane comes back. Tela starts to form 2. Cytokinesis right there. All right, so if you look at this, interphase is called a resting phase because your cell is there. A majority of the time it's in interphase. You've got your checkpoints, you're making chromosomes. That takes some time. Prophase, chromosomes are visible. Your spindle forms as centrioles move. So if you look at this, as your centrioles are moving to each end of the cell, if you look at it right now, you're going to have spindle fibers that are forming. All right, we have to have spindle fibers because, see right there, They have to help move your chromosomes around. Your chromosomes don't have little arms and legs to help them move around. It's your spindles. All right. Metaphase, chromosomes line up along the equator. Anaphase, chromatids separate. So you can see where they call them, that's why I was talking about the different words, chromosome versus chromatid. The chromosome is right here. There's two of them. See that? Now it's a chromatid when there's a single one. All right, the nuclear membrane during telophase, it forms on each side and cytokinesis begins. All right. How is cytokinesis different in plants versus animals? Well, think about that. Plants have what that we don't have. They have a cell wall. So what happens is in plant cells, it begins when a new cell wall forms. So look down here and you can see our animal cell, how it kind of just splits in two. But look at the plant. The plant starts to form a new cell plate, and then it connects and it's a cell wall. So that's how it's different. Whereas an animal cell can just pinch in two, a cell has to form a new cell wall. All right, let's go to the last page of the notes. Okay, the cell cycle in cancer. So this is really a big one. I mean, I feel like we hear more and more cases of people having cancer, and it really gets people talking about, like, what do we do? How do we fight this? How do we make it better? Well, I think to do that, we need more students that understand why it's happening, and maybe then you guys, your generation, will be the ones to really, maybe make some headway in this area. So anyways, we have neoplasm, which is just an abnormal growth of cells. Sounds really southern there. But it's an abnormal growth of cells, right? They're not growing like they normally should. Now here's two words that we tend to hear, and I know some people get them mixed up, or they hear it and they're like, ooh, is that the bad one or is that the good one? Well, benign is the good one. It's the non-cancerous mass of cells. So maybe you have an abnormal growth of cells, but it's nothing bad. That's something we can take care of. We don't have to worry about it coming back. It's non-cancerous. Malignant is where it's cancerous. So that's an abnormal growth of cells that, yeah, is cancerous, and they're going to continue to do that. Now, the cancer definition is a cellular growth disorder that results from the mutation. So here's the big one. It's a mutation of genes that regulate the cell cycle and its cell growth. So what happens is when you mutate this gene and it's messing with the cell cycle, basically your cell cycle does not stop. So normally you go through interphase, mitosis, which is PMAT, cytokinesis, and it stops until it needs to do it again. Well, in this, you get a mutation where it doesn't stop. It just goes right back into interphase and just keeps growing and growing. And that's why cancer cells grow, and that's why you don't want them moving around. Say that you have a cancer cell that starts traveling through your body, well, then it's going to do that. It's going to throw these cancer cells other places, and they're going to keep doing this. All right, so here's the thing. Cancer cells, they lack differentiation, so they don't care. Like, they don't have, when I say that, I mean there's certain cells that are liver cells. There's certain cells that are your small intestine cells or blood cells. Like, they don't care. They don't know what they are. They do not have a normal nucleus. Can't talk here. They form tumors, which are just masses because they continue to go through mitosis until they form these masses. They undergo metastasis and angiogenesis. So I don't know if you've ever heard of like a metastatic tumor where it continues to grow. That's what they mean, metastatic. Is what that is. All right, so let's look at this. We have just some more how mitosis is producing daughter cells. So let's look. We've got, always, we start with one cell. When we get to the end of these daughter cells, we now have two, which I think I'm jumping ahead a little bit here. Let me go to here. Yeah, mitosis, it produces two daughter cells. So that picture back there was just another kind of picture showing you what's happening. We keep seeing them. So mitosis is going to produce two daughter cells. All right, so two identical daughter cells. Now those daughter cells will eventually become just the regular cell, and their cells will form two daughter cells. So the two daughter cells contain the exact same number of chromosomes as the original parent cell. That's what you want to happen. Like I said, now sometimes in sex cells, and other cells can happen too, you get where anaphase doesn't properly separate them, and you get an extra chromosome in a cell, which means another cell is missing one. So you get these disorders in babies where they're either missing a chromosome or maybe they have one too many. So instead of 46 pairs, you have 23 pairs, which would give you 46. You now have 47 chromosomes, or maybe you only have 45. Daughter cells are diploid, which means they have double chromos. All right, haploid means half, so this means you have 23 pairs. If you're haploid, you just have 23. Okay, so diploid means that they have all 23 pairs of chromosomes. Okay, getting there, guys. All right, so HeLa cells. This is really interesting because a HeLa cell, or also called, you can see where it's different, like where they just do lowercase. HeLa cell is a cell type of an immortal cell line. So it's Henrietta Lacks. Is that what Henrietta Lacks? She had cervical cancer, and they, of course, took cells from that cervical cancer because they can do that, and they studied them. Well, the patient who eventually died of her cancer, look at this, in October 4th of 1951, but they still have these cancer cells in a laboratory, and they have studied them, and they use them in research. So it's the oldest and most commonly used human cell line in studying cancer, and there's a book here. I have not bought it. I may have to do that for the end of this year. I keep saying it, but I think I'm going to get on there and buy it and see about that then. There's also an article here if you want to click on it. I'm going to try to get through these notes in one class period, so we will not look at it too much here. Get off of there. But basically, how this was such an impact and such groundbreaking, stuff has come from these cells. So anyway, there's an article there. Like I said, I'm not going to go too much into it. I want to get through this in one class period. There's a cell quiz here. So name the phases starting at the top. If you're going from the top here. So follow along with me. This one right here would be interphase. This one right here is prophase. And it's hard to see, but you can see the X's, and you can see the spindles, or what they call kinetochores. Meta, lining up down the middle. Anaphase, pulling apart, and telophase, which cytokinesis then is right there at the end. All right. Name the phase. So if we're looking at this, they are pulling apart, so I know it is anaphase. X is spindle, or like I said, they will call it a kinetochore. I think I'm going to spell it wrong. Kinetochore. And Y, that is your centriole. All right, your centriole is almost like your team captain. They're on each end, and they're attached to the spindles, which are pulling those chromosomes back. So I think of them as team captains, picking their teams and pulling them to them. All right, name that phase. I know there's some Chinese writing up here, but name that phase. Well, it looks like to me that you're starting to get that indentation of where the cytoplasm starts to close in on itself. You're starting to make two new nuclei. You can see them here. So I would say that this is telophase. Name that phase. Well, what gives it away to me right away is, yes, I can see these chromosomes, but I also see these guys, the kinetochore spindle fibers, and that tells me that it is prophase. All right, and then you have this on your sheet, so I put this on here so you could answer it on your sheet also. So let's look at this. We have, my goodness, it's hard to see these little guys, but we have centrioles. We have got chromatin. Right now, when it's in a jumbled up mess like that, they will call it chromatin. It's the same thing. It's your DNA. It's just when it's in a jumbled mess, they give it a name, chromatin. It'd be like me being like, oh, I'm going to give you a name for each class you're in. You're still, I'm going to pick on Liam. You're still Liam, but what if we gave you a name for biology or a name for English? Still you, but different names. Okay, let's go ahead and label. This is your spindle, or like I said, if you see the term kinetochore, that is what that means. Is that all the labeling I think there is? Okay, so let's start. It's going to go in order. We've got interphase, where it's just a jumbled up mess. Now, when I can actually start to make sense of these chromosomes, it's a chromosome, or what they call chromatids, or just a chromosome. So this is prophase. Just write pro. Now here I can see them lining up the middle. Now, yeah, look, it's the middle. I know that we're used to seeing it this way, but this is still the middle, the equatorial plate. So this is meta. It's just that this cell were twisted a little bit. You can see them up and down. Now here they're getting pulled apart, so anaphase. Here we are starting to split into two, so telophase. And then finally we have cytokinesis. In humans, each cell except sex cells, which we will talk about when we do meiosis, has how many chromosomes? So in humans, how many chromosomes do we have? You can answer this one of two ways. You can tell me, hey, there's 46, or you could have told me that there are 23 pair. Number 11, after mitosis, how many daughter cells are produced? After mitosis, look at this, we have 2. Go from 1 to 2 cells. After mitosis in a human cell, each daughter cell has how many chromosomes? Well, they are identical. They are the exact same as the parent cell, so it is 46, or 23 pair. Remember, you 23 pair because you got 1 pair, or half of yours you got from mom and half you got from dad. That's the original cell that you started out as. How many phases are in mitosis? There are 4. Now guys, we've talked about interphase and cytokinesis, but mitosis is prophase, meta, ana, and tele, and that is it. It is division of the nucleus. Number 14, which phase of the cell cycle? Now look at this, not which phase of mitosis, but cell cycle, which means interphase, mitosis, or cytokinesis is the longest, and that is interphase. That is where your cell spends most of its time. And then 15, during which phase does cytokinesis begin? That begins at the end of telophase. All right. So that is it. If you have any questions, need anything, let me know, and I will get back with you. Have a great rest of your day.